Hi all. My name is Mason Farmwald, and for my computational chemistry project for Chem 394, I chose to look at I chose to do a computational analysis of metallocarboranes as HIV protease inhibitors, specifically a cobalt carborane. So for my intro, human immunodeficiency virus, HIV, can lead to acquired immunodeficiency syndrome, AIDS, when left untreated. We've all obviously heard of HIV and AIDS before. And it is an important virus for current and future study. There's a lot of reasons that go into that, but some of the biggest ones include that HIV is a highly mutating virus. So it mutates up to 10,000 to 100 times thousand faster than known eukaryotic genomic DNA. So it's super, super, super smart at getting around um, both defenses in the human body and drugs that we develop. So that's one reason why we need to keep developing therapies. Another is the fact that development of HIV drugs has led to treatments in other areas like hepatitis C and leukemia. Another great example is Paxlovid, ritonavir, which was developed as a protease inhibitor for HIV, is actually used in Paxlovid for COVID-19 to inhibit cytochrome P450 metabolism in the body. And then one last reason that research is so important is reducing the stigma against people living with HIV, because HIV now is a manageable disease like almost any other disease, but so many people still see it as a death sentence, which that's not what it is anymore. It is one of the most manageable diseases. Obviously, it's not fun to have it, but if people do get it, they can live with it pretty easily. So the life cycle of HIV involves 7 steps. So you've got the binding, the fusion. Then once you're fused, you have to, so HIV is a single-stranded RNA virus. So it takes that RNA and it uses reverse transcription to make DNA. Then it integrates that DNA into the genome, and then it replicates, and then it reassembles itself, and then it buds out of the CD4 cells that it infects. So HIV works by infecting CD4 positive cells, so T cell or immune cells that have the CD4 receptor on them. Those are the cells that infects and hijacks and uses as its assembly factories, basically. Yeah. And study of the life cycle has led to the development of antiretroviral therapy, otherwise known as ART. Some people refer to it as HAART, HAART, highly active antiretroviral therapy. The terms are basically interchangeable. They mean the exact same thing. Yeah, and then I've got this picture here of some key parts of HIV and how it functions. So there are 7 different classes of drugs that we use to treat HIV. And there's 3 different ways that they work. You either have transcription inhibition, enzymatic inhibition, or entry inhibition. So we have non-nucleoside reverse transcriptase inhibitors, nucleoside reverse transcriptase inhibitors. Those are your transcription inhibitors. So they either look like nucleosides or don't look like nucleosides and allosterically inhibit reverse transcription of HIV RNA. Whereas your nucleoside reverse transcriptase inhibitors, they look like a nucleoside base, and then they get incorporated into the DNA assembly or protein production or any step of that life cycle and produce nonsense or stop codons or anything like that nature. Then we've also got entry inhibitors, so fusion inhibitors, CCR5 antagonists, and post-attachment inhibitors. Those all work in different entry mechanisms. So like fusion, if we go back to the previous slide where it talks about fusion, that will inhibit the fusion part of that. CCR5 antagonists, the CCR5 receptor is a co-receptor on CD4 positive cells, and HIV uses both to bind. So if you can use an antagonist on CCR5, you can stop HIV from even binding to the cell. Yeah. And then the last thing that we have is enzymatic inhibition. So those are through both protease inhibitors and integration transfer inhibitors. And like I said earlier, development of antiretroviral therapy has led to the development of further drugs like Paxlovid, which wouldn't have been possible if we didn't have to create ritonavir for HIV treatment. We could have obviously gotten to that point at some point to figure out a way to inhibit cytochrome P450 metabolism in the body, but this research has helped many other areas of drug development and treatment of other diseases that we face. So my project, I wanted to specifically look at HIV protease inhibitors. HIV protease inhibitors work by, so all the ones that currently exist on the market are peptidic, so that means they look like peptides. And they resemble a transition state analog of the polypeptide that HIV protease cleaves. So HIV will cleave this polypeptide into GAG and GAG pol. Then those are the mature versions of the respective proteins. And by inhibiting that cleavage, it forces the polypeptide that is being generated to never get cleaved. So you never get the mature version of the proteins, which cascades down to eventually HIV not being present anymore. These are some of the protease inhibitors that I chose to investigate. So we have saquanavir, ritonavir, darunavir, atazanavir, and lopinavir. As you can see, they all have that kind of peptide backbone going through them. All right here, you can see the nitrogenous base, or the nitrogen part, and then the oxygen part. And you can see all of that inside of those. But you can see how they have different groups attached to them. So that will give them different properties and different ways to get around mutations. Like I said earlier, since HIV mutates so rapidly, we have to design new drugs that can get around those mutations. Like saquanavir has a lot of mutations against it now. And if you don't have wild type HIV means HIV with no mutations. So we have to be careful about what drugs we're giving to people based on which version of, or which genetic mutations their strain of HIV may have. So specifically, when I was coming up with this project, I wanted to somehow incorporate some inorganic chemistry into computational chemistry, just to kind of see what I could get to mesh together with HIV. So we've got a lot of different things going on here. So while established HIV protease inhibitors are great at their design job, new development for HIV drugs is always occurring, and HIV protease inhibitors have not had a new version of a protease inhibitor introduced for over a decade. I think it's 2 decades at this point. So, which is great that our protease inhibitors function that well that we haven't had to make new ones, but it doesn't mean that we necessarily need to stop development of them just because the ones we have now work so well. And so my train of thought was, okay, I could do some investigation of an inorganic complex or something of that nature that could inhibit some nature or some part of HIV. I found that inorganic polyhedral metallocarboranes are able to inhibit HIV protease in HIV in the same, not in the same mechanism, but in the same fashion that the peptidic protease inhibitors can inhibit the protease. And then specifically the one that I was looking at was this cobalt carborane. So this is the structure before optimization, before geometry optimization run computationally. This is it kind of drawn out down here. And then this, I've got HIV protease here with the binding site. So the binding site of HIV is kind of up on this, they call it like the flat parts on the left and the right here of the dimer. And the ligand binds up in this part. So it's found that the cobalt carborane binds in about the same area that the peptidic inhibitors do. So that makes study of it a little bit easier. So to calculate all of my results, I looked at known protease inhibitors and my cobalt carborane in Gaussian to start. I wanted to compare Hartree-Fock and B3-LIP so I could get a good comparison of some ab initio versus some DFT calculations, and kind of compare the outputs of those. For my Hartree-Fock calculations, I use saquanavir, ritonavir, atazanavir, and lopinavir. And for my B3-LIP calculations, I use saquanavir, ritonavir, and darunavir. And then looking at all of my results that I got, I found that essentially almost everything calculated was relatively very similar. As long as you used a big enough basis set, which I use 6-31G double star, so that means the inclusion of both D and P orbitals. And I found that there wasn't too much of a difference between calculated values between Hartree-Fock and B3-LIP. The only thing that I found a large difference in was the dipole moment calculations, which Hartree-Fock found a significantly lower one every time for everything compared to B3-LIP. So that's something worth analyzing and doing future research on why that occurs. And then there was also cobalt carborane. The cobalt carborane I analyzed under PM6. I had to use PM6 instead of PM3 because PM3 is not parameterized for cobalt. So PM6 is. So looking at that, if we compare all the entropy, the enthalpies, the free energy, and the dipole moments, we see that the cobalt carborane is significantly different from all of the other kinds of peptidic inhibitors, which makes sense because it doesn't look like a peptide. It's comprised of boron, a cobalt, and like 4 carbons, whereas our peptidic inhibitors are completely different compositions. So that would make sense that we see completely different thermodynamic properties. So then the second part of my research, I wanted to look at how Autodoc, specifically Autodoc and Autodoc Vena, do in docking sequinavir so as a known ligand to kind of see like, okay, we know how this binds. How do these programs bind in comparison? And so we can see here that the ligand in purple is the Autodoc Vena result, and the ligand in gray/green is the known crystallographic structure of how sequinavir binds. And through Autodoc Vena, this isn't a great result. It's not sitting how it should, which that could play into how fast Autodoc Vena goes. Autodoc Vena has a lot of just built in, the system figures it out for you, whereas regular Autodoc, you do a little bit more of parameterization yourself. So I think that's part of why. Whereas compared to the regular Autodoc 4.2.6 version, I ran 70 conformations of sequinavir to figure out could Autodoc generate the binding conformation that we know crystallographically or something close. So we've got the lowest binding energy on the left, which is not similar at all. It's not even in the binding area, versus the highest binding energy prediction, which is pretty similar. It was in the genetic algorithm, and there were not really any changes made except for doing 70 iterations instead of the standard, I think 30 that it says. So that was pretty good, seeing that there were no setting changes. And then this was the, out of the 70 conformers generated, these were the spread of the binding energy all the way from around 1,000 to almost 8,000. And so I analyzed this one here on the left of the 1,000 mark all the way up to that almost 8,001. And that's what I'm comparing. The one on the left, the lowest binding was this one. The one on the right was that one. So in conclusion, we know that cobalt carboranes have been proven before in vitro to function as effective protease inhibitors. Comparison of my cobalt carborane, GB18 is how it's referred to in the literature. There's other carboranes as well that have other denotations like 21, 40, 80. But comparison of this to the market available protease inhibitors show that they don't have similar thermodynamic traits, though they both inhibit. So it's interesting to see how we have different properties, and yet they can do the same function. So that is worthy of some investigation. And it's also interesting that Autodoc Vena did not produce the known binding conformation of sequinavir. That might need some further fine tuning, or and that could be of both the ligand or the protease. That could need some fine tuning or some torsional changes or that kind of thing. But Autodoc 4.2.6 produced a decently close conformation to the known binding conformation of sequinavir. So doing a little bit more fine tuning, maybe changing a couple torsional allows and not allows of the ligand or the protein might help to get better results. And then finally, I have all of my references here.